post-translational modification. In this video, we will be focusing on ubiquitination. The content of this video will be as follows. We will start with biological system followed by the study question. We will then go through shotgun proteomics and its method in identifying biomarkers. Next, we will introduce ubiquitination and its role in coronary artery disease. Biological system. The biological system that we will be focusing on is cardiovascular disease, specifically coronary artery disease. It is the #1 cause of death according to the World Health Organization. Coronary artery disease is simply the disease of the blood vessels supplying the heart muscle. This is due to the build-up of arteriosclerotic plaque, where its deposition starts at early age. And over time, the plate can harden or rupture, and this will lead to two scenarios. First, it may narrow the coronary artery and therefore reduce the flow of oxygen-rich blood to the heart. And the second scenario would be forming a blood clot on its surface and completely block blood flow. This will ultimately lead to tissue death, which can cause heart failure and arrhythmia. We will now look into the current diagnosis timeline for coronary artery disease. At 2 hours after admission to the emergency department, clinical evaluation is done. This includes electrocardiogram (EKG). If ST elevation is recorded in the EKG, the patient will be directed for treatment. However, there are some cases where patients do not have ST elevation. They are called NSTMI, which is non-ST elevation myocardial infarction. Within 24 hours, patients with NSTMI will go through a diagnosis, which includes checking of biomarker troponin. The troponin complex, which comprises of troponin T, C, and I, is located in the thin filament of the striated muscle contractile apparatus. In the case of cardiomyocyte injury, cardiac troponins are released into the bloodstream. For myocardial infarction, an initial rise in the troponin occurs within 4 hours after symptom onset. If the patient's data show ST elevation on EKG and elevation of cardiac troponin, then these patients will be directed for treatment. The majority of patients, however, are discharged, and 7% of them have been reported to have died. To save this 7%, we would need to find a biomarker which is present immediately after a heart attack. This is so to reduce the healthcare burden that coronary artery disease has. This leads to our study question, which is to identify stable biomarkers which are found in all stages of myocardial infarction, enabling early heart attack diagnosis and treatment. The requirements of potential biomarker candidates are, first, to have a data with good coverage of all proteins present in a sample. Second, is to have enough number of subjects to ensure adequate research. In the search of biomarkers and mechanism of acquired cardiovascular disease, proteomics takes a very important role, specifically shotgun proteomics. Why shotgun proteomics? This is because it is the most unbiased view of proteins, unlike other approaches such as targeted discovery and targeted. Shotgun proteomics refers to a use of bottom-up proteomic techniques to study the whole proteins in a complex mixture such as serum, urine, and cell lysates by utilizing HPLC and mass spectrometry technology. One of the few features of shotgun proteomics are such that shotgun proteomics are able to identify and quantify protein on a large scale and globally with only minimal protein separation needed. It is also ideal to study protein biomarker with shotgun proteomics. But last but not least, it is able to identify and quantify post-translational modification. However, there is no standard method for preparing protein samples in proteomic studies, such that protocols differ in regards to the sample. This is the overview of the proteomic analysis for cardiovascular disease. In the case of the samples given, UB1 and UB2, our sample collection was done such that we were given cardiac tissue as our sample. It is the aortic punch tissue, which is derived from coronary artery diseased patient. For data acquisition, we use global discovery approach, such that we digest the tissue and we get the total amount of protein. For data processing, protein identification was done through electrospray ionization quadruple time of flight. Last but not least, for data interpretation, with the choice of a protein identified, we study the ubiquitinated protein and the interaction that it has, and therefore the effects of this interaction or the phenotype on the cell. Our study ends here, as we were not able to perform any validation studies. The overview of the experiment is done as such that the protein mixture will be first digested by a protease, and the resulting peptides will be separated in HPLC, followed by SMS analysis, which is the ESI quad TOF, to identify the sequence of each peptide. And with the identified peptide sequences, we can then compare with the database, which we can then search for the corresponding protein identity or homologue. As mentioned before, aortic punch tissues are given as our samples collected from 2 different coronary artery disease patients. Tissue lysis is then done by a reagent-based method. However, some components need to be removed for downstream analysis. Despite that, reagent-based lysis method is very rapid, and it is also efficient, such that it enables high protein yield. Next, after tissue lysis, protein purification is done. This can be done using acetone precipitation, which is one of the better methods as it is efficient and does not require many steps. Purification is needed as most of the informative proteins, which are regulatory proteins, are always in less abundant. However, these less abundant protein or important proteins can still be detected more efficiently with proper pre-treatment and handling. After protein purification, proteolytic digestion is done. In this case, in-solution digestion was used by using serine protease trypsin, which is the most common method. In the reduction and alkylation of the cysteine, the disulfide bonds of the proteins are irreversibly broken up, and the optimal unfolding of the tertiary structure is then obtained. This chemical modification allows for protein with a high number of disulfide bonds, the successful identification, as well as the highest peptide yield and sequence coverage. Next, protein quantification is done using BCA assay, which is compatible with samples that contain up to 5% surfactants. For purification of peptides, the reverse phase C18 cartridge was used to isolate peptides and remove salts and buffer. For separation of peptide fragmentation, reverse phase high-performance liquid chromatography was used. Due to the buffer pattern used, the first few flows of HPLC will contain relatively polar peptides, whereas towards the end, the peptides will be relatively non-polar. Peptide identification was done with mass spectrometer by using ESI quad TOF. From this peptide identification, we are able to obtain information such as the mass, charge, and intensity, or the amount of peptides. For data search, the parameters were set as carbamidomethyl as its fixed modification, and for variable modification, ubiquitin at lysine, deamination and oxidation is used as variable modification. The method that we used was mascot DB search, and the protein of choice was chosen by having following two important requirements, which is such that it is a unique protein present in both samples and it also has a good score. Here you can see that both samples have Erlin 2. As seen, Erlin 2 has a good score of 140. From the protein view of mascot, we were able to see that the sequence coverage for Erlin 2 is very low. This could be due to the low abundance of Erlin 2 in the sample. After blasting, we are able to see that Erlin 2 is the exact match with Erlin 2 isoform based on the expectation value. The top score for peptide, which was chosen after locating that lysine, the ubiquitinated lysine is in the middle fragment, is as shown here. So, what is ubiquitination? Ubiquitination is a type of enzymatic post-translational modification. Ubiquitin is a small polypeptide of 76 amino acids. Ubiquitination is accomplished by the coordinated action of three enzymes, E1, E2, E3. E1 is ubiquitin activating enzyme, E2 ubiquitin conjugating enzyme, and E3 ubiquitin ligase enzyme. Ubiquitin ligase. Ubiquitin can be attached to a substrate protein as a single moiety or in form of polymeric chains. Ubiquitin proteasome pathway or UPP or protein degradation is responsible for the degradation of most cell proteins, which includes regulatory, structural, abnormal, and short-lived proteins. They are considered as cell's protein quality control. Only when the ubiquitin moieties are linked through lysine 48 will the substrate then be degraded by the 26S proteasome. The first ubiquitin molecule is covalently bound through its C-terminal carboxylic group to a particular lysine, cysteine, serine, threonine, or N-terminus of the target protein. Linking this to our biological system, saccharinase protein turnover is important in the heart, which is maintained by the ubiquitin proteasome system, UPS, such that the dysregulation of protein turnover occurs in heart failure. Saccharinase-specific chaperones and ubiquitin ligases are necessary for the assembly and degradation of saccharidoprotein and constitute the protein quality control system in the heart. The protein quality control of the saccharin involves the continuous assembly, which is here, and the degradation of the specific saccharomer proteins. This is balanced by the specific ubiquitination and degradation of proteins by the ubiquitin proteasome system. In the example given here is the co-chaperone co-chaperone UNC45 and HSP90, which is required for the assembly of myosin. This involves the balance of this specific ubiquitination and degradation is important, such that it involves specific ubiquitin ligases that place polyubiquitin tails on targets for degradation. In the other example, where both MURF1 and MURFI3 have shown to specifically ubiquitinate and degrade myosin in a proteasome-dependent manner. In the heart, this dynamic process of protein quality control occurs amid continuous use in order to maintain the fundamental construct necessary for contractility. Another function or the second function of ubiquitination is that it is able to alter the protein cellular location, such that a study was done by Linda Hick where she was able to identify new ubiquitin binding proteins that may recruit the caterin-based endocytic machinery in yeast. The third function of this post-translational modification is its ability to affect protein's activity. In a case study done by Scott and his colleagues, it was found that ubiquitinated membrane proteins are able to regulate protein sorting at the level of endosomes and multivascular bodies. Another function which was found is its ability to promote or prevent protein interaction. Various ubiquitin modifications are able to adopt distinct conformation and serve different cellular functions. Within the polycubic ubiquitination chains, ubiquitin can form 8 different linkages types, using one of the 7 intrinsic lysines residue or methionine at position 1. Each linkage type has a distinct three-dimensional topology which results in specific interactions and underlies the regulation of various biological functions.
Hi, I'm Kai Yi, and I will continue to explain how ubiquitination can be associated with ERLIN2. So what exactly is ERLIN2? ERLIN2 is usually associated with ERLIN1, so let's just explain them together. ERLIN1-2 complex acts as lipid rafts in the ER membrane, and it also acts as a recognition factor for activated IP3 receptors. Little is known about the mechanism of ERLIN1-2, but from the ring shape of ERLIN1-2, we can guess that it forms some kind of pore in the ER membrane, or it completely or partially encircles IP3 receptor tetramers. ERLIN1-2 recruits E2 and E3, specifically UBC7 and RNF170, and acts as a ubiquitin ligase and directs transfer of ubiquitin from the ER to the protein substrate. Hence, IP3 gets targeted for ubiquitination and gets degraded by proteasome. ERLIN1-2 is important in ERAD. What exactly is ERAD? It is ER-associated protein degradation which simply acts as quality checks for the ER. As for now, how ER proteins that are substrates for the ER-associated degradation pathway are recognized for your polyubiquitination and proteasomal degradation is largely unsolved. We are only aware that ERLIN1-2 complex helps ubiquitinate IP3 and targeting them for degradation. However, more recently, ERLIN2 has been recognized as a novel mediator of ERAD. ERLIN2 binds to activated IP3s and other ERAD substrates, leading to polyubiquitination and subsequent degradation of these substrates. Hence, we can postulate that ERLIN1-2 complex is a recognition factor that targets IP3s and perhaps other substrates for ERAD. Normally, when proteins are accidentally misfolded, they are targeted for degradation by proteasomes. However, as we age, we accumulate mutations. One of the most studied mutations of the proteostasis pathway is when there is a degradation in the proteasome. Hence, it is unable to degrade the misfolded proteins, thus leading to accumulation. But mutations due to aging can occur anywhere throughout the ERAD signaling pathway. Hence, we propose that ERLIN2 has a mutation. ERLIN2 is part of the ERAD pathway as discussed earlier and may have a potential to be mutated as the proteostasis pathway declines with age. As we know, polyubiquitination is the process of targeting misfolded proteins, which are not functioning properly anymore, for degradation. Thus, results from mascot data search shows that there is actually a mutation or a non-functional ERLIN2 that is being targeted for ubiquitination. Ubiquitination of ERLIN2 can happen by two ways. One, self-ubiquitination by its own ER ubiquitin ligase, or it can also be ubiquitinated by other ubiquitinase in the ER. As you can see from this small diagram, there's actually a mutation in the ubiquitin ligase, and it's unable to ubiquitinate its substrates. Instead, it self-ubiquitinates or modifies itself, but that's just additional information. So the main point we have to take note here is that ERLIN2 currently has a mutation. So what happens when there's a mutation in ERLIN2? It can actually lead to a accumulation of misfolded proteins. ERLIN1-2 complex is important in ERAD as when there are mutations in ERLIN1-2 complex, it will lead to the inability to target misfolded proteins for polyubiquitination, leading to an accumulation of misfolded proteins. These accumulations eventually result in ER stress. UPR is activated in response to an accumulation of unfolded or misfolded proteins in the lumen of the ER. This means that UPR will induce ER stress-associated programmed cell death, apoptosis, to protect the organism by removing the stress cells. In the ER, 3 pro-apoptotic proteins are able to sense increase in misfolded polypeptides and activate the UPR. So these 3 proteins are PERK, ATS6, as well as IRE1. These three proteins are very important in directing the UPR signaling pathway, and we will come back to them later. So now we have to take note that in normal conditions, GPR78, which is a heat shock protein, is actually bound to molecules of UPR sensors. However, when in ER stress, GPR78 will dissociate from UPR sensors that leads to, in turn, an induction of UPR, leading to pro-apoptotic pathways activated in stress cell. This can lead to an induction of oxidative stress, inflammation, as well as apoptotic response. So let me explain to you a little bit about the signaling pathway of apoptosis. When ER stress occurs, GRP78 will dissociate from the three sensors and initiates UPR. PERK signaling pathway will decrease protein synthesis by reducing protein load in ER lumen, leading to an activated PERK that will phosphorylate E1F2-alpha. This process induces cell death via certain gene families activating ATF4 as well as CHOP, which is very important in the signaling pathway of apoptosis. Now that we are aware of how mutations in proteostasis network leads to misfolded proteins that can cause ER stress can actually cause common age-related human disorders. And one of the most common age-related human disorders are cardiovascular diseases. Inflammation is an immunological response to infection or tissue damage and protects the body from such injuries. Chronic inflammation aggravates tissue damage, and it can influence the development of cardiac hypertrophy, heart failure, coronary artery diseases, as well as atherosclerosis. ER stress and inflammation signaling pathways are connected through various mechanisms that can induce cardiovascular disease. We focused on apoptosis pathway earlier, so now let's look at how ER stress can contribute to inflammatory signaling pathway. In atherosclerosis, increased PERK and IRE, TRAF2, and accumulated ROS, which is reactive oxygen species, can actually activate as well as augment inflammation response. PERK and IRE TRAF2 complex can actually recruit IKB kinase, which is IKK, as we see from the diagram, which will phosphorylate IKB, resulting in the degradation of IKB and the nucleotranslocation of NF-kB. The NF-kB-IKK pathway is a key regulator in the induction of inflammatory mediators. Prolonged ER stress observed in atherosclerotic lesions is an important contributor to pro-atherosclerotic progression, which means essentially the promoting of fatty deposits in the artery. ER stress was found in all major cell types in atherosclerosis, including macrophages, vascular smooth muscle cells, which we will call VSMCs for short, as well as endothelial cells. Let's now look at them one by one. Macrophages. Macrophages are necessary for the removal of apoptotic cells from plaques, so that's what they do in their normal function. In early plaques, they act normally, so apoptotic cells are quickly phagocyted by macrophages. In late lesions, however, a number of factors may contribute to defective phagocytic clearance of apoptotic macrophages, leading to secondary necrosis of these cells as well as a pro-inflammatory response. The cumulative effect of all these late lesional events is the generation of necrotic core, which, in concert with pro-atherosclerotic effects of residual surviving macrophages, will promote further inflammation, plaque instability, as well as thrombosis. Thus, the ability, or the lack thereof, of lesion phagocytes to safely clear apoptotic macrophages may be an important determinant of acute atherothrombotic clinical events, which can serve as a potential biomarker. So this is important. Looking closer at the pictorial view, we realize that early atherosclerosis, the macrophages are able to remove the defective apoptotic cells, but in late atherosclerosis, macrophages are defective, and this can lead to the formation of a necrotic core. Endothelial cells. ER stress-induced apoptosis diminishes the barrier function of vascular endothelium, which is also similar to gunshots on endothelium, causing it to become more permeable, and as well as induce phenotypic changes in the endothelial cells that can be directly responsible for increased risk of thrombosis as well as other late atherosclerotic complications. VSMCs. In atherosclerosis, the aberrant proliferation of VSMCs will promote plaque formation. There's an inflammatory response in the vessel wall, thus leading to the disruption of IEL, the extracellular matrix modeling, leading to subsequent formation of a bulge or a distension of an artery. This will cause further cell death as well as vessel wall degeneration, ultimately culminating in IA rupture. So, from this diagram, due to the inflammation of VSMCs, it can lead to apoptosis and a decreased expression of collagen biosynthesis. Thus, your artery will become less elastic, which is what we want to avoid, right? Because it will lead to unstable plaque. This image is a summary of the three factors we have mentioned earlier. The VSMC and endothelial cells get inflamed and deteriorate, leading to an inflammatory response, but in late lesions, when macrophages are unable to remove the apoptotic cells, eventually leads to plaque rupture. Which brings us back to our main point again. Due to a dysfunction, mutation in ERLIN2, leading to instability to target misfolded proteins for ubiquitination, this causes an accumulation of misfolded proteins and eventually ER stress. Thus, an inflammatory response. Currently, very little is known about the exact mechanism of ERLIN1-2 complex and even the degradation of other molecules, which excluding IP3, which is the only concrete evidence we can find. So all that we have mentioned earlier is actually postulations from research data as well as our own generated results. ER stress and ER-induced cell death plays a non-redundant role in pathogenesis of atherosclerosis as well as other complications. We predict that pharmaceutical targeting of novel ERLIN2 as well as pathway mechanisms may be beneficial in the treatment as well as prevention of cardiovascular pathology. More research should be done about the ubiquitination of ERLIN2. As you can see, ERLIN2 only has one PTM, which is glycosylation, hence we conclude that ERLIN2 mutation is actually very novel, and how ubiquitination may affect its role in preventing ER stress and eventually acute cardiovascular event. These are our list of references, and thank you so much for listening to our presentation. Have a good day.